I'm going to set myself a timer here to make sure that I don't talk too much, because sometimes I'll do that. This is actually a really beautiful segue in this session, because it's allowed me to hear more of the basic science and the math and focus on a lot of what I'm interested in, which is then what do I end up doing with it for my patient. So I'm a physician in infectious disease and live and work in Mumbai doing both clinical cohort studies and laboratory studies. And I'm going to be talking about this as a conversation of how these data end up becoming something that we can make use of. So hopefully there's not going to be as much math or as much data presented here, and I'm hoping that it's sort of a simple concept. Nothing to disclose, and I don't need to convince this audience of the importance of advanced diagnostics, but I'm personally fairly enamored of whole genome sequencing as a diagnostic tool, and the reason for that is that as we're getting more information, it's becoming more accessible, more people are using it for a variety of different methods, including epidemiology and drug susceptibility testing, and we know that the cost, while still much higher than, say, a gene expert, is coming down. And for example, where I'm working, the difference in price between doing second-line drug susceptibility testing and whole genome sequencing for the way we do it is less than $15, which is not on the right time frame, but it's not that far off. The UK is already translating this into a clinical diagnostic, so we should be talking about how we're going to end up using this for physicians and for patients. The problem with this conversation is that we're not all speaking the same language. As we know from the last couple of talks, we're seeing a lot of phylogenetic trees, we're seeing epidemiologic data, we're seeing specificity in terms of codons or amino acid changes. The difference between a SNP and an indel may or may not be totally at the tip of everyone's tongue, even in this room. Microbiologists are largely interested in drug susceptibility, and for a clinician's perspective, what I really want to know is, what can I give my patient, and how much can I trust the information that I'm getting from this test? And the reality is that as a clinician, we're used to getting bad information all the time from either patients or family members or the lab, or great information from all those people, and we're trying to really figure out which things we can trust to what degree so that we can make a decision. That's why I have a job. So one example of how this would play out was a patient that I saw about 1.5 years ago who we knew had TB, right? So they had an expert performed, which was great. We knew that they were MDR because they had had first-line phenotypic drug susceptibility performed. It seemed that they were susceptible to ethambutol and streptomycin, but we needed to come up with the rest of the regimen. Second-line testing is still cooking. We were fortunate to have genotypic data back, so we knew that this patient had a mutation in the EMBB gene, which in theory, we were told didn't really matter for this patient, but there was a mutation in the TLYA gene, which is associated with capreomycin. This particular mutation, we didn't really know what to do with at position 84, the arginine to glycine mutation, so I went to PubMed and said, okay, we're seeing that this one doesn't seem to have an MIC fold change when it's been seen in vitro. We don't have in vivo data, we don't have outcomes data, and I just really want to know, can I give the patient the drug? In the end, we didn't have to, but that's not really the point. Looking at what other people have done to try to approach this, I did find a survey that was put out last summer asking a bunch of different providers, mostly in the UK, what information they actually wanted and what information they were comfortable with. This was a small survey, which is not yet followed up, but has some follow-up work being done, where they looked at a good split between clinicians and people working in either epidemiologic or surveillance settings. Most people said they felt comfortable interpreting SNP, so single nucleotide polymorphisms, related resistance, phylogenetic trees, clustering, and SNP distance, which already tells you that the people who you're talking to aren't average primary care doctors, right? So we've talked earlier today about at what point in the field you're going to implement these different kinds of tests. If you have MDR-TB, in Baltimore at Hopkins, if you're unfortunate enough, you might end up talking to me, but you probably won't. You're certainly not talking to your primary doctor. So you're already looking at people who have a certain degree of education about how to interpret these data. Most of them don't want a large proportion of epidemiologic data on the output, but it depends on the person. So over the last year, I've been looking at different reporting methods that have come out of different sites, mostly between Europe and North America, and we see a wide variety of different kinds of information. So there's technical data looking at how much of this is human mapping reads, how much of this is TB versus other things. what coverage we see in the gene of interest, as well as heteroresistance, which is a topic that goes very, very deep, and I'm not going to go into very much right now. Epidemiologic data, mutation-specific data, which can be presented either as a codon or as an amino acid, and then the drug resistance stuff that I'm really interested in, not only referring to drugs of interest, but also how likely is it, based on what we just heard from Paulo, that this particular mutation matters for this particular patient, which always needs to come with that other sense of how much do I trust that piece of information that I'm getting. So what we're going to try to end up saying is some method that will create a report that a clinician like myself can review that's going to balance that technical data set with simplicity and readability. Because if I see you for 15 minutes and not uncommonly you come in with 100 pieces of paper that I have to flip through, what I want to know is quickly, can I or can I not give you this medication? So decisions that end up needing to be made are going to include things like how we make a statement, what we name that statement that we're making, how much to report on these genes or drugs of interest versus everything that we get out of whole genome sequencing. All of that is going to be informed by which analytic pipeline we use. To what extent do we report heteroresistance? Because the reality is that that's hard to find, hard to be specific about, and doesn't necessarily have a clear implication. And then to what extent should we be lumping drugs by class? Fortunately, we have a precedent. So I know that this is a TB audience, but one of the joys of infectious disease is that we also take care of HIV patients. And for those of you who are not familiar with it, the Stanford HIV database is awesome. I get my genotypic report back, and I don't necessarily know, I'm sorry this is a little bit blurry, but for this patient that I saw, if there's an M46I mutation, I don't necessarily know what M is, I don't necessarily know what I is. What I do need to know is that I can take that, that mutation and put it into this online tool, and it'll give me this beautiful report, which tells me that atazanavir is going to have intermediate resistance, but I can still use darunavir, and definitely don't use nelfinavir, not that I would anyway. So this is the standard of care for HIV. I get tested on this in the boards. So there is precedent for using whole genome sequencing, for using next-generation sequencing technologies in clinical practice. So things that need to be clarified. The first is how we actually predict resistance. And a lot of what I'm going to be presenting in terms of our approach to the presentation, the report of these data, is going to come from the RESEC-TB data set. So we're going to be talking about the likelihood ratio approach that Paulo just mentioned. So the little bit of math that I have in this presentation is a likelihood ratio being a sensitivity over 1 minus specificity, which is basically an indication of, given that you have that test finding, how likely is it that you have that disease? This was just discussed a minute ago and much more beautifully than I'm going to, so I'll skip over that. The next question that came up is how we're going to name the mutation. So do we want the nucleotide? Do we want the amino acid? Do we want both? From the RESEC-TB database, what we've ended up settling on is presentation of both. And the reason is that depending on the gene of interest, one of them may be more relevant than the other. If you don't have, if you're just working in the promoter regions, it doesn't necessarily matter what amino acid would be coded for by that set of codons. And in addition, within this data set, certain mutations, despite having the same amino acid change, will have distinct likelihood ratios or confidence in terms of the output. So if we're trying to say with certainty or with some metric of certainty, in position 445 of the RPOB gene, if you go from C to G, you have a much higher likelihood based on our existing knowledge than if you go from C to A. Another question that's going to have to be answered here is, if we're talking about a report, how important is it for me to say that T changed to TTCG, CAT, GCC? You get the idea. That also makes me wonder, to what extent do I need to report inconclusive data? We know that we have new drugs of interest. We know that we can talk about things like the ATPE gene in bedaquiline. We have some in vitro data if you search PubMed for different sites. But if it's outside of the gene of interest, to what extent do you need to present upon that? What we've done in thinking about how to apply a report has been to look at genes of interest that our expert committees have said are things that we should be reporting upon. We've not yet included newer drugs, but this is sort of a room to grow concept, right? It's a continued conversation that is likely to change over time as additional data become available through the RESEC-TB data set. So the same genes that Paulo mentioned are the ones that we're putting into our report. And to the extent that we know it, so say INHA's implication for ethionamide in addition to isoniazid end up going into our report as we have it set up. But for the moment, we have not been thinking that we're going to end up including things like the 133-fold MIC-associated mutation that changes from alanine to proline at site 63 in the ATPE gene. Alternatively, there is room for discussion to say, if I only need 5 drugs, and I know that I've got this, even though I don't know that it's clinically applicable because I don't have in vivo data or epidemiologic data, I might still want to know that, given that there is a caveat. Additionally, we're going to have to wonder about the extent to which we restrict specific reporting pipelines. So the ReSec-TB data platform is particularly useful because you don't have to be a bioinformaticist to use it. It is clear that they've done a good job of setting out that it is TB and not a human and not Pseudomonas that you're sequencing by the time you get the report back. You have confidence in the mutation. But we also don't want to be so closed off that there's only one way to do it because we haven't yet agreed upon that as a world community. So if the ReSec-TB pipeline is going to make sure that I'm dealing with TB, make sure that I've got good coverage and good depth at the site that I'm reporting, there's always going to be a question of how we report and interpret results that come from alternative pipelines. So in this setting, it's going to be important to say in your clinical report which pipeline was used, and then provide the information about which pipeline was used and what that means to the person trying to interpret. There was a good talk yesterday, so I won't go too deeply into this, but the threshold for calling heteroresistance is going to become important. If the ReSec-TB data set is going to say that you need 70% of reads at a particular site to be able to say that you have that mutation available, what do you do when there's a mixed population? This report is from one of the freely available sites on the internet. I know that I'm not showing you a lot of the reports that I looked at, but that's because most of them are confidential or classified. This one is not, and I think it's a really good example to show how one might present differing reads at a particular site. So you can see in that first section there, for S431X in the RPOB gene, there is a minority population where a resistant allele was seen once and a susceptible allele was seen 23 times. And for me, if I don't have good bioinformatics data, if I don't have a good background on all of this, I can at least look at that and say, okay, it's probably susceptible. The extent to which that one matters right now, I'm not sure about. The extent to which 6 months from now, it's really going to matter, I can then deal with later, which is not that different from what I might do for HIV, looking at a variety of sequences over time. But I probably would want to know information like this. And then the last thing is going to be whether to lump drugs by class. So we know that we talk all the time about RPOB gene mutations. We've talked about gyrase A mutations. They're going to be different from quinolone to quinolone. We don't clinically or epidemiologically or as programs use moxifloxacin the same way that we use ofloxacin or levofloxacin. Some people would, but most people wouldn't, switch to rifapentine the same way that they would use rifampicin, but rifabutin is often used interchangeably clinically. So what we've done is we've said that you can probably lump ofloxacin and levo, and you should probably use moxi later, and maybe if you're in Bangladesh, gadafloxacin. And if it's a point mutation that's going to affect rifampin, it's probably also worth referencing what it's going to do for rifabutin, if that's known, and other rifamycins. So as I mentioned, we're trying to maximize that balance between technical data and simplicity and readability, and make it useful to multiple end users, leaving out the really intense epidemiologic data and just focusing on the stuff that's going to be necessary up front with a way to get more information later if you want it. We focus on a 2-page report that currently has 6 parts. The first is the basic patient and lab information that you're going to need in a clinical report, followed by first-line DST and second-line DST, a basic assessment of coverage and heteroresistance to the effect that people want to look at on the second page, reference to lineage, and an explanation of methods. So thinking in terms of a conversation, thinking in terms of translations, we've shown you up front, the first column is for the first-line drug mutations, which drug you're looking at, followed by how to interpret it, which has both the word, the symbol, and the color, so that if you're red-green colorblind, this isn't going to bother you. And hopefully some of these words are going to be interpretable across different languages. And as I mentioned,
The next thing we're going to say is a comment on how confident we are in that statement. So rifampin is resistant with high confidence using Paolo's method. And if you want to keep looking, because you're a very interested clinician, you can see that there's a gene target, an amino acid, and a codon change, and then information about how much this impacts rifampin based on what we know, and how much this impacts rifabutin based on what we know, which might not be the same thing. Similarly, for no evidence of resistance, you're probably good to use the drug, and then possible resistance on the bottom, because we know that there are a very large number of mutations in pyrazinamide, not all of which necessarily matter, most of which are often assumed to matter. So for this one in particular, we said there's insufficient data based on the RESEC-TB data set, and the mutation is known to disrupt enzymatic activity. Likewise, for second-line drugs, the first row is combining ofloxacin based on similar expectations of resistance, but separated out from MOXIE, with different rows out for kanamycin, amikacin, and capreomycin, because we know that we might have different confidence and different importance of mutations in the genes that affect both drugs. And finally, on that second page, we end up referring to the proportion of mapping, number of reads, coverage, and heteroresistant calls within a set of genes that are thought to be important based on expert committees, comment on the lineage, and then explain those same methods that Paolo did a great job of explaining 5 minutes ago. Our next steps are going to be to show you guys basically what we've been talking about and consider this to be a continued conversation so that we can update our reports as we learn more information from other end users. There will be a planned WHO meeting to sort of finalize our reporting template, and this will continue to be updated as clinical data become available and more data happen within the RESEC-TB data set. I'm going to stop there and definitely say thank you to Tim and to Marco for including me in both the RESEC-TB and CPTR groups, and for Fine for helping me to be here to talk to you guys today, and thank you all for your attention.